Oxidative phosphorylation. The process that generates most of the ATP is called oxidative phosphorylation because it is powered by redox reaction. For oxidative phosphorylation, I want you to remember what is oxidative phosphorylation. We're going to discuss later in more detail. Oxidative phosphorylation accounts for almost 90% of the ATP generated by cellular respiration. A smaller amount of ATP is formed in glycolysis and the citric acid cycle, Krebs cycle, by substrate level phosphorylation. For each molecule of glucose degraded to CO2 and water by cellular respiration, the cell makes up to 32 molecules of ATP. I want you to remember there are two types of mechanism to produce ATP in cellular respiration. One is major mechanism to produce ATP. That's the oxidative phosphorylation through electron transport chain inside the inner membrane of mitochondrion to produce most of ATP. The other mechanism is small number of ATP. They are produced by minor mechanism of ATP production. That's the substrate level phosphorylation. Substrate level phosphorylation means some enzyme in glycolysis and citric acid cycle, those enzymes, they go through typical enzymatic activity to produce ATP. That means, as you can see in the picture, for those enzymes active site, they make binding for the substrate with phosphate, and another, the active site, they make bind with ADP, and those enzymes take phosphate group and take over into ADP to produce ATP. So after they produce ATP, those product ATP released out from active site, then another substrate, they bind into the active site and produce another ATP like that. So one after another, one by one, they produce. So small number of ATP, they are produced by this minor mechanism of substrate level phosphorylation. However, most of the ATP, they are produced through that oxidative phosphorylation by electron transport chain in the form of mass production. That's why large amount of ATP, they are produced by oxidative phosphorylation. So I want you to remember two mechanisms for the production of ATP in cellular respiration. One is oxidative phosphorylation. The other one is substrate level phosphorylation. I want you to remember. Glycolysis harvests chemical energy by oxidizing glucose to pyruvate. Glycolysis then means sugar splitting breaks down glucose into two molecules of pyruvate, pyruvic acid. Glycolysis occurs in cytoplasm and has two major phases. Energy invest phase. So in the beginning part of the glycolysis, they invest ATP. And second part, that's the energy payoff phase. That means they're gonna produce ATP. So first phase is they consume ATP, but later on they produce ATP. And that glycolysis occurs whether or not O2 is present. That means, glycolysis, it doesn't matter the presence of O2 or absence of O2. So with O2, without O2, it's just going on. So it is nothing to do with O2, and it is nothing to do with mitochondria, because it occurs in cytosol, in cytoplasm. That's why it is nothing to do with mitochondria and nothing to do with O2 for glycolysis. This picture symbolizes glycolysis. So it is outside the mitochondria, nothing to do with mitochondria, and nothing to do with O2 either. So through the glycolysis, they produce ATP, actually 2 ATP, by substrate level phosphorylation. This one overall calculation for the productivity of glycolysis. In the first phase, they consume ATP, so 2 ATP used up. However, later on, 4 ATP produced. That's why produced 4 and consumed 2, so 4 minus 2. So net productivity, that's the 2 ATP produced through glycolysis. And another thing, that's the NADH. So through the glycolysis, they reduced 2 molecules of NADH. That's the productivity for glycolysis. Don't try to memorize all those numbers. So if you understand just the concept, that's enough. Okay, for glycolysis, in the beginning, we can start from glucose. And as you can see, ATP molecule get consumed to produce glucose 6-phosphate, and this glucose 6-phosphate gonna get converted into fructose 6-phosphate. This fructose 6-phosphate consume one more molecule of ATP, become fructose 1,6-bisphosphate. This fructose 1,6-bisphosphate gonna get split into 2 molecules of C3 You don't need to memorize all those chemical names though. So I'll give you just the idea for the concept of the glycolysis. Two molecules of 3 carbon molecule, they just show just one side, but instead they just put two molecules. So now they reduced 2 molecules of NADH. Now, at the last part, they produce 2 molecules of ATP by substrate level phosphorylation. And just before producing end product, pyruvic acid, they produce another 2 molecules of ATP by substrate level phosphorylation. That's why 4 minus 2 makes 2 molecules of ATP. That's the net productivity for ATP production by glycolysis by substrate level phosphorylation. So end product, that's the pyruvate, 2 molecule. After pyruvate is oxidized, the citric acid cycle completes the energy-yielding oxidation of organic molecule. So through the citric acid cycle, the sugar gonna get completely broken into CO2. So in the presence of O2, pyruvic acid enters the mitochondrion, where the oxidation of glucose is completed. So that organic compound completely broken into CO2. Before the citric acid cycle can begin, pyruvate must be converted to acetyl coenzyme A, which links glycolysis to the citric acid cycle. So the pyruvic acid gonna get broken one more time, become 2 carbon molecule acetic acid, and that acetic acid bind with coenzyme A to become acetyl coenzyme A. And this step is carried out by a multi-enzyme complex that catalyzes 3 reactions. So before entering into citric acid cycle, pyruvate gonna get oxidized further. This one showing you pyruvate in the presence of O2, they're gonna further oxidize there. So one CO2 released out and become 2 carbon molecule acetic acid with binding coenzyme A to become acetyl coenzyme A. Just before that, NADH gonna get reduced from NAD plus form. The citric acid cycle, also called the Krebs cycle, completes the breakdown of pyruvate to CO2. So through the Krebs cycle, organic compound, they're gonna broken down completely into CO2. The cycle oxidizes organic fuel derived from pyruvate, generating 1 ATP, 3 NADH, and 1 FADH2 per ton. So, as I mentioned earlier, FADH2, they are very similar electron delivery man to NADH, but their ATP productivity little bit lower than NADH. That's another coenzyme. This one showing you the productivity of citric acid cycle, Krebs cycle. That actually the molecule join into the citric acid cycle, that's the acetyl coenzyme A, that join into the citric acid cycle, coenzyme A released out BAP. Then it can go through the citric acid cycle. They reduced 3 molecules of NADH and 1 molecules of ATP, that's the substrate level phosphorylation, and FADH2 get reduced from 1 molecules of acetyl coenzyme A. That's why if we start from 1 molecule of glucose, they produce 2 molecules of pyruvic acid and 2 molecules of acetyl coenzyme A. Then for NADH, they reduce 6 molecules of NADH because they produce from 2 molecules of acetyl coenzyme A and 2 molecules of ATP by substrate level phosphorylation and 2 molecules of FADH2 through the citric acid cycle. So you have to make it double if you start from 1 molecule of glucose. The citric acid cycle has 8 steps. Never mind 8 steps. Each catalyzed by a specific enzyme. So all those biochemical reactions, they are enzymatic biochemical reaction. The acetyl group of acetyl coenzyme A joins the cycle by combining with oxaloacetate, oxaloacetic acid, forming citric acid. The next 7 steps decompose the citrate back to oxaloacetate, making the process a cycle. That's why we call cycle. And the NADH and FADH2 produced by the cycle relay electrons extracted from food to the electron transport chain. So the citric acid cycle, they produce ATP by substrate level phosphorylation. However, high energy electron, they reduced NADH and FADH2 electron delivery man. Then those NADH and FADH2, they deliver electron into oxidative phosphorylation, for electron transport chain for oxidative phosphorylation. Acetyl coenzyme A join into the citric acid cycle, Krebs cycle, by binding with oxaloacetate, That's the 4 carbon molecule. So 4 carbon molecule, 2 carbon molecule, they producing 6 carbon molecule citric acid. There's 6 carbon molecule. And it goes through the citric acid cycle, the Krebs cycle. Then we can see on the right hand side NADH reduced, and near the bottom you can see another NADH is reduced from NAD plus, and CO2 one released out and another CO2 released out. We can see it. That's why we regard that organic compound is completely broken down into CO2, because 2 CO2 released out from a 1 molecule of acetyl coenzyme A. That's the 2 carbon molecule. That's why for the citric acid cycle, that organic compound is completely broken down over here, with 2 molecules of CO2. And continue to reduce FADH2 and another molecules of NADH. Then the maleic acid, malate, that's the 4 carbon molecule, turn back into oxaloacetate, oxaloacetic acid, to begin with another molecules of acetyl coenzyme A that make up the cycle, for citric acid cycle or Krebs cycle. The last step to produce most of ATP is the oxidative phosphorylation.